So this week we're talking about metabolism. Has anyone in here taken biochem yet? Okay, you have? Okay, have you gotten here yet at all? Okay, nice. Yeah, so this is like a very quick crash course of what you'll do in biochem for like half the semester. Reminder that my office hours are tomorrow 2:30 to 3:30. There's my email. Yeah. Okay, so first I just wanted to zoom out for those of you that are like unfamiliar with metabolism. What it is, like big picture, is how our bodies use the nutrients that we get from food to form ATP, to power all the functioning in our body. And at the same time, we need to maintain homeostasis or regulated conditions when we're like kind of making chemical changes in the body to produce ATP. At the same time, we need to maintain homeostasis. So as described in the lecture videos, another way to describe this is balancing the amount of energy that we take in, and which is food, and then balancing the amount of energy that we let out, which is used either to produce energy, to make ATP, for physical activity, and then also for thermoregulation or maintaining body heat. So there are two really important types of reactions. I would star this slide. Understanding these reactions is really important to kind of understanding everything that we're going to talk about in metabolism. So anabolic reactions are building smaller molecules into bigger molecules. So you use ATP to do that. So an example of an anabolic reaction would be converting glucose into glycogen for storage. So glucose is a simple sugar, and then combining a bunch of glucose molecules together into glycogen, a bigger molecule, is an example of an anabolic reaction. The opposite, a catabolic reaction, instead involves breaking larger molecules down, and we release ATP when we do that. So going back to the other example, this would be breaking glycogen down, a big molecule, into glucose. And in this case, we'd release ATP. So a trick to remember the difference is for a catabolic reaction, you could think of like a cat on a ledge knocking down something, like a vase that shatters, like catabolic. Big to small. Does this make sense? Does anyone have any questions on the types of reactions? Okay. Getting into glucostatic theory. Glucostatic theory basically says that metabolism is regulated by the amount of glucose in your blood. So you want to maintain the amount of glucose in your blood at a certain level, or homeostatic level. When we eat food in the fed state, our blood glucose goes up, and then it becomes hyperglycemic, which just means high glucose. This causes the anabolic reactions to kind of turn on. Like you have high glucose in your blood, so you're going to be building a lot of molecules to store it. And at the same time, when your blood glucose is high, we're going to have satiety signals stimulated. Basically telling your brain, like, okay, we're fed, we can feel full. On the other hand, the fasted state is when you haven't eaten in a while, your blood glucose goes down, which is also known as hypoglycemia. So you have low blood glucose. In this case, the catabolic reactions are going to turn on because we're going to want to break down all those big storage molecules into smaller ones to then bring the blood glucose levels back up and to generate energy. In this case, when we're hypoglycemic, low blood glucose, hunger signals are going to be stimulated. And then interpreting the graph that I put on this slide, when the blue or the hunger line is up, you can see that the blood glucose levels are low. So it's just showing that hunger and satiety are directly related to blood glucose levels. So just to summarize this slide, glucostatic theory therefore refers to the fact that metabolism is regulated. The reactions of metabolism are regulated by the level of glucose in your blood. Okay, so now I was going to do a little drawing that connects the catabolic and anabolic reactions to all of the reactions in metabolism that you should know. So I associate anabolic with blue. I don't know, because I feel like if you're building something up, it might be blue, whereas catabolic is red, because you're like breaking it down. So I'm going to follow that here. So starting with anabolic reactions. Okay, so when metabolism is favoring anabolic reactions, does anyone remember if we're in the fed or the fasted state? Yes, because you have high blood glucose, so you can kind of build molecules up for storage. So we're in the fed state when we're doing these reactions, which is also known as the absorptive state. Okay. So then the first reaction we're going to talk about is glycogenesis. And the second one is lipogenesis. Okay, so before I tell you guys or write on the board what these two are, does anyone have any guesses what these would mean? We're in anabolic fed state, and this is a reaction called glycogenesis. Yeah.
It's formation of glucose and formation of like triglycerides. Right?
Yeah. So just be careful with your wording.
Oh, like glycogen. Sorry.
Yes, correct. So I think it's really helpful to break down the terms. Glyco is referring to glucose or sugar. Genesis is like to build. So in this case, we're making glycogen, which is a bigger storage molecule from glucose. So we're going to write glucose. To glycogen. And then same goes for lipogenesis. Now we're talking about lipids and we're building something, genesis. So in this case it's free fatty acids, to triglycerides, which is the storage form of lipids. For each of these reactions, there are certain enzymes involved in the cascade of events that forms glycogen from glucose or triglycerides from free fatty acids. The two mentioned in the lecture videos I'm going to write down here so you can associate the certain enzymes with the certain reaction. So in the case of glycogenesis, we have hexokinase, and glycogen synthase. This one, glycogen synthase, is easy to remember because glycogen synthase making glycogen. You just kind of have to memorize that hexokinase is involved in forming glycogen in glycogenesis. In the case of lipogenesis, there's lipoprotein lipase. Or LPL. Okay, now moving on to the catabolic reactions. Which is in the fasted state. So again, in the fasted state, our blood glucose is low, so we're going to want to break down these storage molecules, the triglycerides and the glycogen, to increase our blood glucose, and so our body can use the triglycerides and the glycogen to form energy. So now on the other side we have glycogenolysis. And lipolysis. So again, if you analyze kind of like the way the word is built, glyco refers to sugar, lysis means to break. So glycogenolysis therefore means to break down glycogen into glucose. Lipolysis, again, lipo means lipid, lysis means to break down. Like you can think of like lysing a cell, like breaking down. So now we're going the opposite direction, breaking down triglycerides into free fatty acids. then I will write down the enzymes that are mentioned so you can associate with them with a specific reaction. Here we have glycogen phosphorylase. Glucose 6-phosphate. I think those are the only two you actually have to know for this one. And then over here we have hormone. Sensitive lipase. Okay, so after the catabolic reactions, we've increased, that's not glucose. We've increased the concentration of glucose and free fatty acids in the blood. Does anyone know the process that glucose can then go through to make energy? Yeah. Did you say it? Yeah, perfect. So then, big connection is that glucose then enters cellular respiration in your cells to make energy. So then this glucose is going to go to cellular respiration. To make ATP. And the first step of cellular respiration is glycolysis. So you got three pretty big glycoterms: glycogenesis, glycogenolysis, and glycolysis. So glycogenolysis is glycogen to glucose. Glycolysis is glucose to pyruvate. Which is just an intermediate of cellular respiration. And then you have the TCI cycle and but you don't get into the details of cellular respiration, but those are the three main parts to know. And then the free fatty acids cannot enter into cellular respiration to make ATP. They instead have a different process to make energy. Does anyone know what that's called? Okay, so free fatty acids will be used in beta oxidation. To make energy. I guess I'll stay with this one. So when the body runs out of glucose stores, or here, I'll start by saying this. The only type of molecule or nutrient that can enter into cellular respiration is glucose. Glucose has to enter, form pyruvate, go into the citric acid cycle, and then the electron transport chain. However, when the body runs out of glucose, it needs to be able to use the free fatty acids and then also protein to make energy and so that it can enter cellular respiration. When the body is out of glucose, free fatty acids and proteins go through a process called gluconeogenesis, which is another big gluco word. So I'll put that over here. And in gluconeogenesis, we're taking fat and protein and making it into some sort of intermediate that can then enter cellular respiration to form ATP. So this is when we're really out of glucose. We're taking fats and proteins and converting them into an intermediate like pyruvate, or another one is acetyl-CoA. So that they can enter cellular respiration to form ATP. So the way you can remember this one is I would look at the word neo. Neo usually means something like new, or making something new from fats and proteins so that they are able to enter cellular respiration. And then genesis, we're forming a new molecule because we're out of glucose. So I have that here. I put glycogen synthase twice. I believe it should be in glycogenesis, but I'm gonna have to check that. And then I also put gluconeogenesis under the catabolic reactions. It's not a catabolic reaction. It's technically an anabolic reaction because we're building something. And then I also put this note here, the way we broke down the words. Again, lysis means to split or break down, so that tells you it's a catabolic reaction. Genesis means to make or build, so that tells you it's an anabolic reaction. And then glyco always means glucose or sugar associated, and then lipo means lipid or fat. So like we talked about with cellular respiration, here, after glucose is made, it enters the blood. It enters cellular respiration to make energy. And then I just put that diagram there to kind of jog your memory because I'm sure that you've talked about it in other classes. I think you just need to know the basic outline of cellular respiration, that there's glycolysis, then the citric acid cycle, and the electron transport chain. And the purpose is to make ATP. Something else I want to highlight from this diagram is right here is pyruvate. Pyruvate and acetyl-CoA are just different forms that glucose goes through when forming the energy. So in gluconeogenesis, when we use fats and proteins to make these two intermediates, they just enter the cycle right there. So if we formed pyruvate with gluconeogenesis, it just enters right into this part of cellular respiration. If we formed acetyl-CoA with either fat or protein, it just enters right here. Just because glucose is the only thing that can enter glycolysis at the beginning of cellular respiration. So going off of that, glucose is the preferred fuel because it can enter cellular respiration directly from the beginning. Gluconeogenesis, again, is converting the fats or proteins into pyruvate or acetyl-CoA that can then enter cellular respiration. Another note is that gluconeogenesis only occurs in the liver. So we can add that. So the liver converts the proteins or fats into these intermediates, and then they enter the blood, where they can enter cellular respiration. And this explains why glucose is the preferred form of energy, because it's so easy to use. It can just enter right into cellular respiration. This is kind of an exhaustive list. Actually, I don't know if I'd call it exhaustive, but this is a good list of hormones from this unit. So 2 SIs ago, I went over insulin regulation. Insulin is an anabolic hormone, so it's triggered by high blood glucose. And it promotes the formation of glycogen. So when you have high blood glucose, your body releases insulin to help you uptake that glucose into cells and form glycogen. Glucagon is the opposite. It's the catabolic hormone that's triggered by hypoglycemia to break down glycogen into blood glucose. So if you wanted to, we could add let me grab this. This process would be triggered by glucagon, and this process would be triggered by insulin. Insulin is anabolic, helps you form glycogen storage molecule. Glucagon, catabolic, helps you form glucose. Another hormone that's important is leptin, which is known as the obesity gene because it's often misregulated. It's secreted by fat cells to increase satiety. And it also shuts off orexigenic neurons, which we talk about in the hunger and satiety response soon. VIP is vasoactive intestinal peptide, and this one relaxes smooth muscle and the pyloric sphincter, indicating that there's food in the stomach. So if we're relaxing smooth muscle, it kind of just tells you that there's food in the stomach is full, which stimulates satiety. And that's done by anorexigenic neurons. Ghrelin is stimulated or secreted when the stomach is empty. That's supposed to say orexigenic neurons. It stimulates hunger. And then another point to remember from last unit are the enterogastrones that inhibit gastric emptying. So that's CCK, secretin, and GIP. So that's to like slow down digestion. And it indicates that like you have food in the system. I know this can be kind of overwhelming to look at, but this will be good for studying to go back to because you can just kind of keep all the hormones straight. It's kind of why I made that slide. Okay, so this is where the hunger and satiety regulation comes in. The arcuate nucleus is a region in the hypothalamus in the brain that is responsible for regulating hunger and satiety. And the way that it works is that there's a nerve that goes all the way from the arcuate nucleus in your brain to the gut. And it kind of like it gives information to the arcuate nucleus about if we should be hungry or full. So like when the gut is distended, when there's food in the gut, then the nerve sends a signal to the arcuate nucleus and says that we're full. And there are two neurons that do this. The anorexigenic neuron, when that one fires, it stimulates satiety. So the anorexigenic neuron would stimulate satiety if the gut was full, if it's distended. The orexigenic neurons stimulate hunger, and that would happen if the gut is like empty. And the next bullet points will be explained better during this drawing, so we'll just do that. This is like the last slide before we have some top hat questions, so I'll take off this. Okay, so this is just going to be of the arcuate nucleus. So I'll actually put the hormones back up so you can look at them when we draw. Okay, so for this drawing, I'll do red to be associated with satiety, and blue is going to be associated with hunger. So, two hormones, insulin and VIP. We're just going to label this as like the first case scenario. And then also the enterogastrones. Well, we don't need that. Secretin and GIP. Those all stimulate anorexigenic neurons. Well, actually, I'm going to write. So far we have insulin which is released when you have high blood glucose, VIP, which is released when your gut is full, and the enterogastrones, which are released when to slow gastric emptying, so you know that you have like food going through the system, stimulate anorexigenic neurons. Anorexigenic neurons are gonna eventually lead to the release of leptin, which cause satiety. So there's one more intermediate step. The anorexigenic neurons stimulate the paraventricular nuclei, which then releases leptin. And leptin is a hormone, like we have up there, which then causes satiety. So it's kind of cool how it works, because at the same time that insulin is helping your body form glycogen to be stored, it's also stimulating your brain and telling it to release this hormone to make you not hungry anymore, which is cool. The opposite, when you are hungry, ghrelin is released, and that's the hormone that's secreted when your stomach is empty. Ghrelin stimulates orexigenic neurons. And what orexigenic neurons do is they inhibit paraventricular nuclei. So it's going to prevent the release of leptin, which then makes you hungry. So this means inhibit. And then that kind of indirectly leads to hunger. And basically, just to draw it back, arcuate nucleus just refers to these two neurons. This marker doesn't work. That basically just feed back to this region of the brain. So these two neurons innervate the gut and can then communicate with the paraventricular nuclei to either release leptin or inhibit its release. Does anyone have any questions on that? Yeah.
Is glucagon also in the blue?
That's a good question. I don't have it in the blue in my notes, but that would make sense if it were to be in the blue. But if I don't have it in my notes, I'd assume that he doesn't mention it in the lecture video, which means you probably don't have to know. So then going back to this slide, now it makes sense that anorexigenic neurons stimulate satiety by stimulating paraventricular nuclei that release leptin, while orexigenic neurons stimulate hunger by inhibiting the paraventricular nuclei, so we don't release leptin. And then here's the drawing, so you have it on the slide too. This is the last thing I wanted to mention. I just remember this coming up and me not being prepared for it to come up. so I wanted to just throw it on the slide. Note that the spleen and pancreas are only like on one side in the left upper quadrant. Spleen, pancreas. Yeah, the spleen and the pancreas are in the left upper quadrant while the gallbladder is in the right upper quadrant. Something just keep in mind. Okay, let's do some questions. I have a lot of questions. I don't know if we'll get through them all. Okay, first one. What's an example of an anabolic reaction? Perfect. Lipogenesis. So I erased it off the board, but if you remember, gluconeogenesis. Oh, it's technically an anabolic reaction. Yes. So that one would also work. But the best answer here would be lipogenesis. Which hormone stimulates hunger? Nice. So if you look at our drawing, all the rest of them are involved with satiety. Which of the following does not stimulate satiety? Nice. Yeah, so this one was a little tricky, so that was good. Yeah, you guys got it. So this one is from last week, which is why it has some responses. I just hadn't gone over it yet, so I moved it down. The arcuate nucleus is a region of the hypothalamus that controls what? Leptin release, signifying what is stimulated by insulin, inhibited by ghrelin. So this whole drawing tells you that the arcuate nucleus is involved with hunger and satiety, and then leptin release signifies satiety. Where is the arcuate nucleus? Yeah, perfect. So the arcuate nucleus itself is located in the hypothalamus. It does have neurons that innervate the gut, but the arcuate nucleus is in the hypothalamus. What are the two populations of neurons and their functions in the arcuate nucleus? And you can select two. Yeah, you guys got it. I feel like I've drilled that, so keep going. I didn't explicitly go over this, but it's in the lecture videos. Why is leptin called the obesity gene? Yeah, you guys got it. They have mutations in the gene causing them to produce less leptin, which leads to less feelings of satiety. Which of the following is a reason that lipostatic theory that states that fat produces signals that modulate eating behavior to maintain a particular weight incorrect? So why is lipostatic theory incorrect? Yes. So this question, I didn't go over lipostatic theory either, but it's just kind of saying that the body's metabolism isn't based off of fat, it's based off of glucose, blood glucose levels, glucostatic theory. A good way to approach this question would be by process of elimination. So we know that lipostatic theory is not true. It says it in the question. And leptin cannot be the only hormone that controls hunger and satiety because we have all these other hormones that are involved with controlling hunger and satiety too. According to glucostatic theory, what does the fed state activate? Anabolic or catabolic reactions? Yeah. High blood glucose initiates forming storage molecules, building. What is the process of making glucose from non-carbohydrate sources, or protein or fats? Yeah, perfect. Neo, making something new. So we can form pyruvate or acetyl-CoA to enter the cellular respiration to form energy when there's low glucose availability in the body. What are the two primary energy storage molecules in humans? Can you select two? No? Okay. Yeah, I just realized, like, I didn't see people putting in 2. So just select 1, and then we'll talk about the other one. This was a good question. Okay, so you guys are pretty sure it's glycogen, which is a storage form of glucose. Does anyone know? So glycogen is one. Does anyone know what the other one is?
Okay.
Yeah. So the second one is triglycerides. The primary functions of sugar and fat molecules is like energy storage. Proteins are more for like, I think of them as like doing things, which isn't like scientifically correct to say, but sugar and fats are for storage. And the storage form of glucose is glycogen, and the storage form of fats is triglycerides. What is the primary molecule used for fuel in metabolism? Yeah, you guys got it. It's glucose. I don't think I asked this. Oh, okay. I was just gonna say this out loud, but I asked it, so. True or false? Free fatty acids can be used by the brain for fuel after glucose stores are depleted. Okay, so you guys are pretty split on this one, but it's actually false. The brain cannot use fat as an energy source. The brain can only use glucose. I'm not sure why, but I don't remember why, but that is something that could come up. Where does gluconeogenesis take place? Yeah. Gluconeogenesis is in the liver, and then it releases those intermediates so they could be used elsewhere in the body for cellular respiration. Yeah, that's all I have for you guys. Feel free to come to office hours tomorrow or email me if you have any questions before the exam. And good luck. You got it. Is this the second to last one?
Yeah.
Yeah, only 2 more. You're so close.